right, thanks everyone for joining our webinar here. We've got a three-part webinar mapped out for you with the following topics. I'll be introducing the BioCyc website and database collection and the Pathway Tool software. And then I'll be giving more of a scientific talk about the metabolic reconstruction process. How does it work when we infer the metabolism of an organism from its sequenced genome? What is the algorithm that we use for that? And then I'll be giving a very short introductory tutorial to the BioCyc website. Then Suzanne Paley will be giving a tutorial to the omics data analysis capabilities within the BioCyc website. And Jeremy Zucker will be talking about AI-assisted metabolic and regulatory investigations of the circadian clock in the fungus Neurospora crassa. And I want to say a big thank you to ASBMB for putting on this webinar. So with that, let's get started with the introduction to BioCyc and Pathway Tools. So BioCyc is both a web portal and a database collection. So it's an encyclopedic reference on genomes, pathways, and regulations. That's where the psych in BioCyc comes from, stands for encyclopedia. We right now have 18,000 total databases. Most are microbial, but we cover important model eukaryotes and humans as well. And of those 18,000, 60 of the databases have been manually curated with their contents derived from 130,000 publications. Now, the main way to access the BioCyc databases is through the biocyc.org website, which supports a number of different use cases, such as information seeking, finding information about particular genes or pathways or metabolites. It has capabilities for analysis of transcriptomics and metabolomics data, which is what Suzanne will be talking about. And it also has comparative analysis capabilities and some capabilities to support metabolic engineering. Now, what is Pathway Tools and how does it relate to BioCyc? Well, Pathway Tools is the software that we use to create new BioCyc databases, and it's the software that powers the BioCyc website. So when you are interacting with biocyc.org, you're interacting with the Pathway Tools software. It's also an enterprise pathway genome data management system, which means that, let's say your lab is sequencing a number of genomes, you can use Pathway Tools to analyze those genomes and to store the data locally in your organization. The software runs actually in two different modes. It runs as a desktop application on your Mac or Windows or Linux machine, or the same binary program can also run as a web server, as it works for the BioCyc website. And so one way to use Pathway Tools is to use it to install a private set of BioCyc databases within your own local network. But one of the main reasons for installing Pathway Tools is to create BioCyc-like databases, or pathway genome databases, as we call them, PGDBs, from your own genome data. And the software lets you edit, update these databases to, say, add new gene functions and pathways. It lets you build quantitative metabolic flux models using flux balance analysis. And overall, it supports all of the same use cases as does the BioCyc website. So a little more detail about BioCyc. Again, 18,000 databases. All the databases share the same types of information. They all cover pathways, reactions, substrates, the proteome, and the genome of the organism. And then we divide the databases into 3 tiers to reflect their quality levels. The tier 1 are very highly curated databases, some of which have received person decades of curation. The tier 2 databases are moderately curated. Usually they've received under a person year of curation. And then the tier 3 databases, the rest are uncurated, they're computationally derived. And when we create a new BioCyc databases, here's the process we follow. We take an annotated genome from the NIH RefSeq component of GenBank, We then apply various computational inferences to the databases to infer new information, and we'll go into this more deeply in the rest of the webinar. We perform quite a few different data import functions to import data from other databases like UniProt. And for the tier 2 databases, we'll then perform manual curation for some of the databases. What do we mean by curation? Curation basically means manual updating of the databases, mostly by people with PhDs in the life sciences, who will correct computational inferences, incorporate information from the experimental literature. They'll author what we call mini reviews about genes and pathways and add citations to the literature. And they'll add new pathways, new gene functions. So we're really trying to centralize in one common place the huge amount of information that's being published on organisms before and after their genomes are sequenced. And another way to look at pathway tools and BioCyc is that once you create a pathway genome database with pathway tools, it becomes a multi-use database that can be mounted on your website as an online encyclopedia for an organism. It can be queried computationally. We can derive metabolic models from the database, and we can create zoomable metabolic maps and omics data analysis tools from the database. But the database is really at the core of all these functions. All right, let me now take a minute to do an intro BioCyc tutorial. And I'm going to go to biocyc.org. And I'm just going to highlight a couple things here on our home page. One of the really key concepts about BioCyc is we have 18,000 databases, but most of the time you're only searching one of the databases. And here it's telling me that the current database I'm searching, which is the default when you first connect to the site, is E. coli K12. If you want to change the database you're searching, like let's say I want to change to Bacillus subtilis, I click on that link, this button here, change current database, and I'll enter, I can enter a genus name, like I can type Bacillus, and we'll see all the Bacillus genomes here. I can type Bacillus subtilis, and we'll see the subtilis genomes. Or I can type, if I know what strain I'm after, I can just type a strain name, and here I'm getting all the organisms that have 168 in their name, which happens to be the strain name for B. subtilis. So I can click on that and then click OK. And now that is my current database. And I can enter a search like tryptophan and hit return there. And what you'll see is that the results are organized according to the type of object that I've matched. So we get pathways that contain tryptophan in their name, proteins containing tryptophan in their name, RNAs with tryptophan in their name, and metabolites, Go terms, and reactions. So this sorting gives you some insight into the rest of BioCyc, namely, we have multiple types of information pages in BioCyc. We have reaction pages, we have metabolite pages, we have gene protein pages, and we have pathway pages. And I'm not going to show you all those right now, but you can experiment with this on your own, enter a search string like tryptophan and try clicking through some of these. I'll just show you real quickly the metabolite page for tryptophan. Aha, it wants me to log in, okay. So here's the B. subtilis metabolite page for tryptophan, which has some basic chemical information about tryptophan, synonyms and SMILES and INCHI string, links to other databases. And notice these tabs here, which will take me to additional information. Like if I click reactions, this shows me all the reactions in B. subtilis that consume tryptophan or produce tryptophan. and what pathways they're present in. And I can click on the reaction to go to a reaction page. I can click on the name of the pathway, if there was one shown here, to go to the pathway page. Everything here is clickable. So that's a real brief introduction to BioCyc. Susanna's going to show you more, and I'll mention later on, actually, I think right now, I'm going to mention that we're going to hold a follow-on tutorial in another week or so on June 29th at 11 a.m. And Susanna's going to put the URL in the chat or the QA section if you want the full URL, or you can write down the small one. And we're going to go into some of the more advanced search capabilities, the genome browser, comparative genome browser, a really powerful tool called Smart Tables. We just don't have time today to give you tutorials on all these different issues, but we'll go into more detail on the 29th. And we also have online webinars on a lot of these capabilities on the website as well. Okay, so given that introduction, I'll give you another minute to write this down. I'm now going to talk about metabolic reconstruction. So let's go on to that. Okay, and before I do that, this is an overview of all the different software tools in BioCyc and pathway tools. And again, this is why we can't cover it all today. Today, we're going to be covering metabolic reconstruction, one of the pathway informatics capabilities. We'll be covering omics data analysis. Jeremy will be talking about metabolic modeling, but we won't have time to talk about a number of the genome informatics tools or regulatory informatics tools. Okay. All right, let's now talk about metabolic reconstruction. That's one of the core inferences in BioCyc and pathway tools, is that we're going to infer the metabolic reaction network of an organism. And so let's just start out with a statement of what the metabolic reconstruction problem is. The problem is to infer the qualitative metabolic reaction network and metabolic pathways of an organism from its annotated genome. So really the key input is the sequence genome of the organism. And in particular, it has to already be annotated, meaning gene positions have to have been called by software, and protein functions have to be predicted for a good fraction of the genome. Gene calling and function prediction are not capabilities of pathway tools. So that's required as an input. And the input can be a fully assembled genome, or the genome can be in contigs. The software can work with either one. The other main input is a reference pathway database. In our case, we use BioCyc as the reference pathway database, and I'll explain what BioCyc is in a few minutes. The outputs of this process are a list of the predicted metabolic reactions and pathways for the organism, and those are created in the new pathway genome database for the organism, as well as reports in the form of HTML files that list the inferred reactions and list the inferred pathways and the evidence for those pathways. Now, put another way, here's the basic information flow within pathway tools. You feed in an annotated genome. It creates a new pathway genome database. And once you have this database, the database can then be searched and visualized and analyzed using the navigator tools. The navigator is one of the main components of pathway tools. The editor tools can be used to update the database. And then the Metaflux tool is what's used to create metabolic models from the database, quantitative metabolic flux models. Now, why is metabolic reconstruction important? What do we get from it? Well, for one thing, it's a first step toward creating a quantitative metabolic flux model for the organism. You need to know, excuse me, before you create a quantitative metabolic model, you need to know qualitatively what reactions and pathways are present. And also another reason to infer the metabolic reaction network is, and pathways, are that pathways are more mentally tractable units that correspond to biological phenotypes. In other words, a cell typically has on the order of several hundred pathways, which is much easier to wrap one's mind around than several thousand genes. Another reason why metabolic reconstruction is useful is that pathways guide us to search for missing enzymes and identify misannotated enzymes. And I'll give you two examples. Let's say we infer a pathway, but enzymes are only present in the genome for 4 of the 5 steps in the pathway, the dark blue steps here. Well, that's telling us that this 5th enzyme is probably there somewhere. Now, it may not be there. It could be absent from the genome and inactivate the pathway, but it tells us this is something to look for more carefully in the genome, because most of the time, such enzymes have been overlooked by the genome annotation pipeline. Conversely, let's say that in a pathway of 4 steps, only one of the 4 steps has an enzyme present. Well, another interpretation is that this enzyme was misannotated by the annotation pipeline, and that it may code for this step right here, because it's kind of unlikely that the organism would have only 1 step out of 4 and retain an inactive pathway. As you'll see, another useful aspect of metabolic reconstruction is that inferred pathways can be used for analysis of high throughput data using the techniques that Suzanne is going to tell you about. And finally, pathway inference fills gaps in the metabolic network. If you're going to build a quantitative metabolic model, you need a fully connected metabolic network in which you have fully connected paths from the input nutrients to the end products of biosynthesis. And in many metabolic modeling systems, they don't do pathway inference. They only infer reactions. And so this blue reaction would be missing. Whereas when Pathway Tools does a metabolic reconstruction, when it imports this pathway, it also imports this missing reaction as well. So even though the enzyme isn't present, the reaction is, and gives us a more fully connected network. What are some of the challenges in metabolic reconstruction? Well, one is that genome annotations contain many errors, so they can throw off the metabolic reconstruction. Genome annotations also often omit information like EC numbers that makes it harder to do reactome inference. And I'm going to skip this one. Another challenge is that there are sometimes multiple variants of a given pathway present in our reference database, Metacyc. For example, we have something like 10 variants of the TCA cycle. And so choosing the correct variant to infer can be challenging. And in general, as Metacyc has grown over the years, we've had to make the inference algorithm more accurate to cut down the number of false positive pathways. Now, I've mentioned Metacyc a number of times. Metacyc is a metabolic encyclopedia. It has kind of a special place in BioCyc in that it's the one database in BioCyc that does not describe one organism. It describes metabolic pathways, reactions, enzymes, and metabolites from all domains of life. We're basically trying to capture in Metacyc an example of every known metabolic pathway that's been experimentally studied. And Metacyc has been derived from 66,000 publications, so it's a really solid backbone of reference pathways to use for predicting pathways in other organisms. And here's some statistics on Metacyc. We have about 2,800 pathways in Metacyc, 16,000 reactions, and about 16,000 metabolites. And if you look across all the pathway pages and enzyme pages and add up all of the mini reviews that are found there, the textual summaries of pathways and enzyme function, you'll get almost 10,000 textbook page equivalents. And I'll compare Metacyc to KEGG. The first line is actually comparing BioCyc to KEGG. The number of genomes, number of organisms is about 3 times larger in BioCyc. This is from an earlier version of Metacyc, which is why the numbers differ a little from the previous page. KEGG contains very few literature citations, whereas Metacyc contains 62,000. We have a much larger number of pathways if you compare to the modules in KEGG, which are the equivalent sized pathway type in KEGG. Metacyc has about 40% more reactions. KEGG does have more metabolites than Metacyc does. And KEGG is largely missing the mini reviews that Metacyc has. We couldn't figure out how to count them in KEGG, but in general, KEGG contains very few mini reviews. And here's some information about the taxonomic distribution of Metacyc pathways. Most are found in bacteria. Plants are next most frequent, followed by fungi, metazoa, and archaea. These don't add up to 2,900 because a given pathway is sometimes found in more than one of these major divisions. All right, now how does metabolic reconstruction actually work? Well, there are 2 main phases to metabolic reconstruction. The first is reactome inference. And so what we do in reactome inference, the goal is to infer all of the metabolic reactions catalyzed by the enzymes of the organism. And to do that, the software considers every protein in the annotated genome, tries to infer the reactions it catalyzes using the following steps. It tries to match the protein functions to Metacyc reactions using the following techniques. It first of all looks to see if there is an EC number in the input annotation. And if there is one, we simply look that up in Metacyc, and the EC number will identify either a single reaction or sometimes a handful of reactions that the enzyme catalyzes. If there are gene ontology terms provided in the annotation, which is kind of rare, but sometimes they are provided, those can be looked up in the gene ontology database, which often map to EC numbers and then to reactions. And finally, the software considers protein names, enzyme names in the annotation. Enzyme names like threonine synthase, carnitine-CoA ligase will map. If we look those up, Metacyc has a large number of enzyme synonyms. So we can simply look up these names in Metacyc and they will map to a metabolic reaction. Now, there are some protein names that are kind of high-level, non-specific names like putative electron transport flavoprotein. Those will not be mapped to a reaction. And non-metabolic enzyme protein names like for leader peptides, those will not be mapped to reactions either. I do want to mention that in a lot of annotations, there will be additional words in the protein function that make it more difficult to match the protein name to a name in Metacyc. Because if we look at a name like putative carbamate kinase alpha subunit, really the core enzyme function is carbamate kinase. And so we've developed a tool that will prune away all these extraneous words that I've shown here in gray to leave behind the core enzyme name, which then we look up in Metacyc. Now the full annotation is kept in the final database, but for matching purposes, if we strip off this extraneous information, that gives us a lot more reliable matches. Finally, if we still have not gotten any match for this protein, we will look up the gene name if one is provided, because gene names can be used to identify metabolic reactions as well. And so finally, if a reaction is inferred for the protein that we're considering, the software will import the reaction and its substrate molecules from Metacyc into the new PGDB that we're constructing. So that's the reactome inference phase. At the end of this phase, We've imported anywhere from several hundred to a few thousand metabolic reactions into the new database we're building. And we then use those metabolic reactions to perform pathway inference. Now, one of the reasons we rely on Metacyc is it's the most extensive microbial pathway database. And so what the pathway inference algorithm does is for each pathway in Metacyc, we compute a score that expresses the likelihood that the pathway is present. And the score integrates the following information. It indicates what fraction of pathway reactions have enzymes present in the genome, ignoring spontaneous reactions, which of course don't require an enzyme. So if we have a pathway of 5 reaction steps, if every reaction has an associated enzyme present, then that pathway is going to get a very strong score. We also can, however, we weight the score given to the component reactions by the number of pathways a given reaction is present in. In other words, if we have one reaction that's present in 10 pathways, and that can happen, then that one reaction will provide very little evidence for the presence of any one of those pathways. The score also considers whether a pathway is outside its designated taxonomic range. Metacyc assigns, the curators assign likely taxonomic ranges to a given pathway. So in other words, if a pathway is designated as a plant pathway, but it will get a lower score if we're trying to predict it into a bacterium. There's also a notion in Metacyc pathways of key reactions, which are reactions, if all of the key reactions have no enzymes present, we will not predict the pathway. And so a common example here is the Calvin cycle, which shares a lot of reactions with the TCA cycle. And so in order to predict the Calvin cycle as present, this this reaction has to have an enzyme. Otherwise, it will get a very bad score. And then we also use some special rules to distinguish variant pathways, especially for TCA cycle and glycolysis, which have the largest number of variants. And so I'm going to go through this fairly quickly. You can review this in the recording if you wish, but in general, we're computing a pathway score as a value between 0 and 1. And the pathway score is the sum of the reaction scores, where divided by the number of reactions plus a boost if the organism is within the taxonomic range of the pathway. Then we compute reaction scores as the sum of the presence of an enzyme for that reaction, plus a boost from a uniqueness score if the reaction is present in only a single pathway, plus a key reaction score if this reaction is a key reaction of the pathway. And finally, once the score is computed, we then use the following rules to actually decide if we accept or reject the pathway. And I'm not going to read through every one of these, but for example, there are some types of pathways that we just don't perform prediction for. So if the pathway is, say, a transport pathway or a metabolically engineered pathway, we're going to reject it. If the pathway has all its reactions present, and if it's outside its taxonomic range, we will accept it. We're going to reject it if it's outside its taxonomic range or if it's missing enzymes for all the key reactions. And I'll note the ordering between these two rules is important. We can accept reactions outside their taxonomic range if all the reactions are present. And finally, we will include a pathway in the prediction if its score exceeds the threshold that we've defined, the user has defined. And otherwise, the default decision is to reject the pathway. And I'll note that pathway tools can perform all of the same processing for metagenomes as well as for genomes. there are really two ways to process metagenomes. One is to bin the metagenome data into and create metagenome assembled genomes, or MAGs. And the software can create separate pathway genome databases for each MAG. And we have a whole set of functionality for analyzing MAGs and searching across multiple MAGs, for example. Or conversely, you can use treat the metagenome as one large data set and create a PGDB for it, and even compute the abundances of the pathways present in the metagenome. This is the pathway evidence report that Pathway Tools produces at the end. It lets you view each pathway and see how many reactions does it have, how many have enzymes present, etc. And the colors show us which steps have enzymes present and whether the enzymes are unique to that pathway. And so what do you get once you've done a pathway prediction? Well, you get the ability to browse through the metabolic information space of the organism, to look at pathway pages and look at metabolite pages, as you saw before, look at reaction pages. So you can navigate through the metabolic information space for the organism. We also have a tool called the Metabolic Network Explorer, authored by Suzanne, actually, that lets you start at a given metabolite and expand one reaction at a time to kind of, again, explore the metabolic information space. In this case, I've started at arginine and then navigated to agmatine and putrescine and spermidine to see what reactions can take me. I can follow outward from arginine, and we can navigate backwards as well. The software will also generate these full metabolic network diagrams, which Suzanne will be talking about in more detail, that provide another way of exploring the metabolic network of the organism. And finally, we can create these diagrams for a whole community of organisms. So here we have 8 different metabolic network diagrams for the altered Shadler flora, showing how some metabolic exchanges going on between these organisms. So let me now summarize the availability of Pathway Tools in BioCyc. The 2 main databases in BioCyc, EcoCyc and MetaCyc, are freely available to all. The rest of BioCyc, there's a certain level of free access provided, but we do charge subscription fees to sponsor our curation on a nonprofit basis. Pathway Tools software is free to nonprofits and academic institutions and government labs, but we do charge a fee to commercial organizations, and the software runs on Mac, Windows, and Linux. And I'd be happy to take questions. And Suzanne, do you have any?
Yeah. So there's quite a lot of questions.
Okay.
I'm not sure that we're going to be able to get to them all.
And let me just mention, we'll probably, hopefully have some extra time at the end that we can address unanswered questions.
I've been answering a few of them as we go along, but someone wants to know, are there specific requirements for the annotation tool that we use to annotate? There are actually several questions about how we annotate or what we recommend for annotation.
Right. I mean, some of the annotation tools that we know work well with Pathway Tools are the NCBI PGAP, Prokaryotic Genome Annotation Pipeline, RAST, and PROCA. And lots of our users use those different tools. Let's see. But in general, it's really helpful if the tool produces EC numbers, because those will give quite reliable reaction inference. Not all of those tools do for every gene, so that's why we don't rely on EC numbers, but it's good for the EC numbers to be there. Yeah, I guess I forgot to mention the formats that we accept. We accept GFF3 format as an annotation format, GenBank files, in particular the GBFF file format is a variant of GenBank format. The nice thing about GBFF format is you can pack multiple replicons and contigs into one file. So it just makes it very easy to input that into Pathway Tools, so we highly recommend GBFF format. More issues related to annotation, Suzanne?
Let's see, there was a question about whether we include algae and if we handle algal annotation.
We do. We've applied the software to a number of different algae, and in fact, if you do I think we've set up a homepage called algae.biocyc.org. You can look under the sites menu at the top of the BioCyc homepage. So there are quite a few algal databases available.
Can mass spec, well, that's more of an omics question. Can I use this to create a new pathway inside a bacteria that doesn't have it? Oh.
Well, you can use the pathway editing tools to define a new pathway that is not in Metacyc. If it's one that you're hypothesizing, yeah, you can create any pathway, any reactions that you want in the database.
A question about integrating it with Python or automating the process.
Right. So a couple angles there. We do have a batch version of the pathway inference. And so some of our users will run thousands of genomes through the software. You can set things up such that just by running pathway tools from the command line, you can process one genome. So set up 1,000 different genome input files and you can process thousands of genomes. And now in terms of APIs, we do have APIs in Python, Lisp, Java, Perl, R, and we have a web services API. So there are lots of APIs available to both query the data in a PGDB and to update the data.
Someone asks, why are transport reactions rejected?
Well, it's transport pathways that are rejected. We don't predict those. Probably just we haven't really gotten around to fully thinking through how that's going to work. I will note that we have a tool for inferring transport reactions from transporter names. So that's another one of the tools in Pathway Tools.
Someone asked, is it possible to create a community metabolic model from metagenome using Pathway Tools?
Yeah, you can. So the approach would be to bin your metagenome into multiple mags, create PGDBs for each individual mag, and then the Metaflux modeling tool, which Jeremy will be talking about, it can create models for individual organisms or for organism communities.
Someone asks, Peter, why did you specifically choose to work on putting BioCyc together? And what is the end state of BioCyc? And what do you hope it enables?
Well, great question. I don't know. I've just always been interested in integrating scientific information and looking at the big picture. And it's always been interesting to me how, journals are this great form of scientific communication, but the information about, say, E. coli is just scattered across tens of thousands of journal articles. And to me, it's just much more information, much more interesting to put all this information together in one place to look at how the whole cell functions. That's kind of what's driven me over the years, is looking at the big picture and asking how can we put all this information together. What's the end state? I don't know. We're always thinking of new things to add to BioCyc, and we always want to hear your input. But what's the final end state? It's probably modeling. It's probably adding a modeling capability, so that, which we've done to a degree, but being able to turn this big picture view of an organism into a model that you can make predictions from, that's one of the end states, I think. And we should probably move on now to your talk, Suzanne.
Yeah, there's loads and loads of questions and they keep coming, so.
Okay, we'll try to get to more questions at the end, but so let me now introduce my SRI colleague, Suzanne Paley, who we have worked together for a couple of decades, and Suzanne has authored a huge number of the tools within Pathway Tools. So when whenever you're interacting with BioCyc, you're interacting in a big way with Suzanne's work. So thanks for all your contributions over the years, Suzanne, and take it away here.
So, so I'm going to be talking a little bit about our omics analysis tools today. We have a whole, and part of it is going to be, there'll be a few slides and then it'll be mostly demo. So we have a whole suite of omics analysis tools, and I am not going to be able to cover them all today, but the first class of analysis tools are the pathway-based visualization of gene expression and metabolomics data. So this involves mapping genes or metabolites to pathways and then overlaying them on our various pathway visualizations. This can be at the highest level, the entire metabolic map of an organism, or on individual pathways, or in between, in groups of pathways, either a tool we call pathway collages, which is an assembled diagram of selected pathways, or tables of pathways that are considered perturbed. So that's one class of data analysis visualization tools that we use. An alternative is the omics dashboard, which is a graph-based visualization, and it uses hierarchical systems of cellular function. And I'm going to be covering both of those today. I'm probably not going to get to these other ones, but I'll mention them briefly. We also can do enrichment analysis. So if you have a set of genes that are upregulated or whatever in some experiment, we can map those to enriched pathways or to GO terms or to regulators. If we have a set of metabolites, we can match those to enriched pathways. We also have various ways to do search for metabolites using monoisotopic masses from mass spec data, metabolite translation services. We can map data onto the genome browser, such as if we have chip data. But I'm mostly going to be focusing on these first two options today. So the first thing people need to know is how you input your data. So what does the data file format need to be? And we take tab delimited text files. So if you have an Excel spreadsheet, you would need to save it out as tab delimited text. The first column of the data file is what contains the identifiers, the genes or the proteins or the metabolites. And those can take any one of different forms. For example, it can be the gene accession. It can be a common name or synonym. Anything that we have on one of our pages as listed as a name, we will accept that. Hopefully they'll be, unambiguous ones are better. We can have links to other databases prefixed by their database ID. Some examples here show some KEGG links or Uniprot links. They actually do not need to be prefixed by the database ID if the links, if the IDs themselves are unambiguous. But some databases have IDs that are just numbers, and there you really do need that. You can use the internal BioCyc identifier, which is available on our BioCyc pages. And then there are various other aspects for metabolite data. We can use chemical formula, monoisotopic molecular weight, INCHI, or you can generate a combination of all of these. For example, this example here is spermidine, but we also has a KEGG link and a PubChem link. So if we have a metabolite that maybe you're not sure if the name is sufficiently unambiguous, or we may have KEGG links for some and PubChem links for others, but not full coverage, then you list a few things and hopefully we'll find something. So that's the first column. Then after that, there will be one or more numeric data columns, and there can be other columns intermixed there that we ignore, but you will specify which are the numeric data columns that you want the tools to look at. And data columns, the data in them can be either what we call absolute data. So these are values like counts or intensities where the values start at 0 and then go in one direction, in the positive direction only. And then we have relative values that are things like ratios or log ratios, where you have a scale that has some center value, which is either 0 or 1, and then proceeds in both directions. And we treat those a little bit separately, so we need to to know what kind of data you're providing. So I'm going to switch to demo mode now. If this will let me. Because these tools are better off being shown, I think, than just talked about. So this is our BioCyc homepage. The tools menu contains all sorts of different tools. Under the omics dashboard is here under analysis, but I'm going to start from the cellular overview, which is also the metabolic map as listed here. So if I, and my current database is E. coli right now. Yes. So this is our, we call the cellular overview diagram or the metabolic map. It's a schematic of all the pathways in the organism. In general, we have biosynthetic pathways on the left here. We have energy metabolism pathways in the middle, degradation pathways to sort of the middle right, and then on the far right are reactions that have not been assigned to pathways. And then around the edge is the schematic involving the double membrane, which we showed transporters. So I can mouse over anything to see what I'm looking at. I can scroll to zoom in, and as I zoom in, more information becomes available up to the very largest level where I can see the actual enzymes. There's lots of things I can do with this diagram. And they're listed in the operations menu here. So there's various highlights that I can do, which I'm not actually going to go into today, but you can explore the various highlighting operations that you can do. So if, for example, if I want to search for a particular compound or a particular gene, or all genes that, like if I want to upload a file and search for all of them, I can do that. I can even do species comparisons in here. But what I want to talk to you about is the use of this diagram for overlaying omics data. So if I want to upload data from well, there's multiple options. I can upload data directly from a file. I can import it from Geo, which is a repository of gene expression data sets. I'm not going to be able to talk about our smart table facility today, but you can save data there and then import it from there. I'm going to start by uploading data from a file. And let me actually quickly show you. So this, the help icon tells you, well, there's a link to help here, and then the individual fields are described. But this will give you, this will tell you how to find more information about the file format. But let me just show you quickly an example here. This is randomly generated data to illustrate your the file format. These are examples of accession number, gene name, BioCyc identifier, links to other databases, and multiple criteria. And we have a similar set of examples for metabolites that show that the set of possible options. So you can use these as a guide. In general, the links to the other databases only work where we actually have those links. So we don't have those for all databases and all genes or proteins. But anyway, I'm going to go back and upload a file. So this is my tab delimited file. It's a time series. So there's multiple data columns. So I need to specify the first data column. I need to say what kind of data it is. These are genes. This is transcriptomics data. This is a data set where we are, it shows the response of E. coli to exposure to hydrogen peroxide, and it's a time series. So now I'm going to choose a set of columns that I'm interested in showing. And this is quite a long time series. I'm only going to extract a few of the columns. And then I need to say whether my data is relative or absolute. In this case, it's relative data. These are log ratios. Because they're log ratios, that's a zero-centered scale. If they were just actual ratios, then it would be well, this is described here, then it would be a one-centered scale. So then I have a choice of options here. I'm not going to have time to go through all of these options. So right now, I'm just going to show this on this diagram, but I've actually preloaded all these other ones as well. So I can briefly show you these. So let me submit that, and it should be quite fast. But if not, I can always switch to a preloaded tab. Here we go. So what it's showing now, so this is the first of the columns. It turns out there are 5 of them, I guess. So I can switch to a different column to show the color. It's colored everything based on, it's colored the reaction arrows based on the genes that that are associated with those reactions. But these can be a little bit hard to read. So I'm going to increase my edge thickness here to make them a little easier to see. And this control panel over here tells me what the color scale is and shows me the distribution of the data. So most of the data is somewhere in the middle, with some of them on the outskirts. And I can play this as an animation and see things change over time, or I can individually look at individual steps. I can, if I mouse over something, I can see a graph of this particular gene, or the 2 genes associated with this step, and see how they change over the course of the experiment. But as I said, this can be a little bit hard to follow, so I can actually customize this display in various ways. For example, there are multiple color schemes option that are available. So I'm going to start by decreasing the number of colors to 5 colors now. And then I can actually tweak where these threshold cutoffs are. Let me make this symmetric. So when I drag this, it's now dragging both ends. So now you can sort of see the numbers of the red is going to capture quite a few more. The purple is going to capture quite a few more. And then the stuff in the middle is the stuff that's really not changing. So it's not that interesting. So let me just hide that by clicking on that. And so now things are really, let me move this a little bit. There we go. So now I get a really good view of what is actually turned on and off in this situation. And now if I play the animation, I can really start to see things changing. There's various other things I can do. I can change the preferences in various ways, especially with the omics pop-ups. I can change them, for example, to an XY plot, or to a heat map. I can show multiple of these at once if I'm interested. I guess this reaction has a lot of genes, but so I can see what they're all doing. So this is the cellular overview omics viewer. Let me just show a few of our other options before I move to the omics dashboard. So this is the table of perturbed pathways. So this text here describes how we compute a pathway perturbation score for a data set. And then we pick out you specify how many of the top scoring pathways you want, and this is the list of pathways that it considers most highly perturbed in the course of the experiment, with additional information about them. And so you can read about this if you wish. This table does not really work for if you have absolute data. It really only works if the data you're supplying are ratios, I believe. Another option is the pathway collage. So this is a set of pathways. It's actually the same set of pathways as from here, but now they're arranged in a customizable diagram. So I can actually edit this in various ways. I can move things around. I can play with the various global properties, etc. But I'm not going to go into more detail about this. I can save this. I can export it to an image file if I wish. This helps produce publication quality diagrams. Probably helps if I move things around a bit. But so I can customize this in various ways. So I get publication quality diagrams. But I'm not going to have time to cover that either. What I want to move on to now is the omics dashboard. So let me show you how to go to the omics dashboard. So, well, let me start here. Let me go back to the home page. So as I mentioned, under the analysis menu is the omics dashboard. And what this is, it organizes cellular systems into hierarchical systems of cellular function. And it's going to, I think it's going to be easier if I start from an example, which I have here. This is my same data set uploaded to the omics dashboard. And let me just, so we have a set of biological systems. So for example, biosynthesis, degradation, energy metabolism, the central dogma, which covers transcription, translation, etc., regulation, various other cellular processes. And within each of these categories is divided into more specific sets of functions. So for example, within biosynthesis, we have amino acid biosynthesis, nucleotide biosynthesis, etc. Within cellular processes, we have the cell cycle, cell death, locomotion, bacterial response, etc. So what we're actually looking at here, so you see this key, we had 5 data columns here. So each of them is color coded with a different color. These are the column numbers that I loaded. They were columns 5 through 9. So these are the colors. Let me increase the size of this. For each of these plots, there's a lot going on here, but we take all the genes that are involved in amino acid biosynthesis, and we, in this particular time point, and we're showing them all here. So each of these little dots is a single gene. The big dot in the middle is the average expression value for all the genes in the system. So you can quickly see at a glance for all the genes in the system, how the average is moving and what the distribution of genes is amongst all the genes in the system and how that's changing. So in general, things go down or stay the same more or less and then go up. And other systems may behave a little bit differently. But amino acid biosynthesis is very broad. It doesn't tell me very much about the details. But if I look at that and decide that I want more detail, because this looks interesting to me, I can click on this and it will give me a new plot. And now this is a big plot, a big plot of amino acid biosynthesis with individual plots for each amino acid. And some amino acids have more genes involved in their biosynthesis than others. So there's just one here, whereas there's a larger number here. But again, this is the same kind of plot where each of the individual data values, each of the individual gene values are small dots, and then the average is also plotted. So then if I want to drill further down, for example, this seems like it's changing quite a lot. I click on this further, and this brings me down to the lowest level where I'm looking at the individual genes with the labels down the bottom. And then this is the graph of how each of these genes change in the course of the experiment. And some of them change a lot more than others, but you can see that the general trend is fairly clear. From here, I actually have several options if I want to dive into more detail. Well, let me actually first, I can actually show the pathways involved here. And so here's a chart of the pathway. This is the histidine biosynthesis pathway with the omics data overload in the pathway context. I can also show the operons that are involved here. So I can see how these are organized on the genome. And it turns out all these genes happen to be on the same operon, which might explain why they are moving more or less in sync, with perhaps one exception here. I'm not quite sure what's going on here. So I can do this for, I can then go look at a different pathway and or different category and look at that. Other options, I can export this graph as an image. I can show the data as a table just for this graph, and then download it if I wish. I can also show the regulators of these genes, and this is only for databases that have genetic regulation in them. So, for example, BioCyc has a lot of genetic regulation, but most of our tier 3 databases do not. But this shows me, these are all the regulators of these genes, and so we see how their transcription changes over the course of the experiment. And then I can select, I can filter this in various ways. For example, if I only want to see what is regulated by this particular gene. And so I see there's just these two, and these genes are also involved. So that's very powerful. There's lots of ways to interrogate this data. Let me clear these. I can customize this in various ways. I can change the scale. I can do various things. I can sort these in various ways. I can show sums instead of, or not instead of, in addition to the actual values, I can add all the values within a category, which can show sort of magnified effects. That's what these bars are. The same data is here, but now it's compressed. And so the big bars are the sums. If this is already log data, if it were not log data, I would have an option to convert it to log data. There's various other display preferences here. So these to be set on a global level rather than an individual level. I can also select which of these series to show or reorder them. I can also use this for an enrichment analysis. And if I have the right data, and let me actually switch to a data set that has this data. So this is a different data set. It's data from E. coli, showing the anaerobic to aerobic transition. What we've actually uploaded here is there's a whole bunch of columns. So there's 3 replicates for every time point. So I was able to tell it to create groups, and it would average these replicates. So I tell it which sets of which input data columns should be averaged together into which group. I can name these groups, which I've done. And this data set also contains additional significance columns because in this case, a significance analysis was run. So these are p-values, which I'm not displaying right now because I can see if we look here, I'm showing the actual data, but not the p-value data. But then what I can do is I can so for each of these data columns that I'm showing, I can assign a significance column. So for example, this particular column goes along with this data column. And then I can run an enrichment. What an enrichment analysis is I'm looking at each of these systems and saying, giving it a score, saying how likely is it that this set of genes would be in my enriched class or in my significant class versus chance. And so I'm doing, my score is a negative p-value, so higher scores are better. Sorry, negative log p-value. So I'm going to run this enrichment data and produce a slightly different display here. And there are other ways we can handle enrichment data. We can use enrichment data with our smart tables facility, which I'm not going to have time to get into. So here's the enrichment data. For each of these subsystems now, we have a total enrichment score for the system. Increase the size of that. A total enrichment score for the system, and then you can this additional bar shows the enrichment score for the highest value subsystem within it. So even if not much is going on within the particular system as a whole, like in this case, the enrichment scores are quite low, but there's at least one subsystem within there that has a high enrichment score. So if I click on that, then I see that it's gluconeogenesis in particular that has the high score, and that's sort of swamped out when it's averaged with the others. But so that alerts me to the fact that this is something that's probably worth clicking on here. And I think I'm actually not quite sure what time I started with this, but I think I'm probably about out of time. So I'm happy to take questions. We do have a whole set of other omics analysis tools that I mentioned, but you're going to have to explore those on your own or look at our help information. We also have webinars on these topics, so you can look at those. So I'm happy to take questions.
Great. Let me make a couple comments. So a number of people are asking about how to obtain the slides from our presentation, and I will send that into the chat shortly, a way to get the slides. And I'm also going to put in the chat info on how to get some of the main publications on BioCyc and pathway tools. Okay, now one of the questions is, can one change the names of data columns to their original names, like control treatment, etc.?
Absolutely. So I'm not sure if you're with respect to which tool. In the omics dashboard, you can absolutely change the columns by editing the labels here, and that will let you do that. Within the cellular the omics viewer here, I'm actually not. I don't think there's a way to set the column names within this tool, but you can actually supply them. The data file has a format that you can specify. If the first column of the data file starts with a $, then we will use those that as a header row. The first row of the data file starts with a $. We will use that as a header row and take the labels from there.
Okay. Another question. There was a question about multi-omics data. How can you import multi-omics data and see the results of the combined data sets?
We don't really have a convenient way of doing that right now. That's something that we are planning on addressing in the future, I think. If you can combine all the data in a single file and load it and look at it that way, but that presents problems. For example, if the scales aren't identical, then things can be confusing. You could, if you have, say, transcriptomics and proteomics data, you could have multiple columns where one applies to the other, and then you could view it in an animation form. So there are ways that you can show this data, but they're not very satisfying. We'd like to address that further in the future.
Well, I think I'd put it a different way, which is like, if you want to combine, say, transcriptomics and proteomics, you can combine it in one data set. I mean, in one input file, as you're suggesting, Suzanne. You could use, say, alternating columns for transcriptomics and proteomics. But as you did say, it's important that you map the values to the same numeric scale because the color assignments, you want kind of consistent color assignments. So you need to think about mapping the data in that way. You could also combine transcriptomics and metabolomics data in one file. But again, as you said, you do need to worry about the color scale and consider mapping the data to a common value range. Okay, is this tool functional with LCMS MS proteomics? I mean, really, the tool should be functional with any data. The tool doesn't care about the underlying technology. It's just mapping the data value ranges that you provide to a color scale.
So long as you provide identifiers that we can map to something that we know about. So, you know, with monoisotopic MS data, if you have monoisotopic masses, we can map those to compounds, or if you actually have IDs.
Right. Is it possible to identify metabolites from mass spec data directly? No, our software does not do metabolite identification. Is there an instruction manual or training class on BioCyc? You want to go through some of the webinars and help materials, Suzanne?
Sure. So if you look at our homepage, there's a whole set of webinars down here that describe some of the capabilities. So there's this whole section on omics data analysis that talks about I believe these are related to the cellular overview, and then these are related to the omics dashboard. And I didn't really have much of a chance to talk about pathway collages, but that was the multi-pathway diagram that I described, or that I showed briefly. And there's a webinar on that. And there are other webinars here. So this is from the homepage, but it's also under the help menu from any page. Get to the webinars link. And there's a website user guide that if you don't want to watch videos, you want to read about it, then you can read about stuff there. And this gives more information about for example, the omics data file format, if you don't remember everything that I've said here.
So Suzanne, you do have a few more minutes. And one of the questions is there a way to batch retrieve or download gene information? If you get a list of genes from a pathway, can you get the information about all the genes? And maybe you could give people just a quick intro to smart tables, like some of the special smart tables.
So, so smart tables are a set of what we call knowledge spreadsheets. So for example, if I have a, this is not show. If I have a set of genes, for example, and I can create columns that are based on various properties. So for example, for this gene, I've added a column for the set of pathways that these genes are involved in, the accession, the sequence, but the set of properties are here. So for example, I can add any other, any one of these properties that I wish. For example, I don't know, the set of synonyms. Well, yeah, set of synonyms, for example. And so I get this new column here. That has the synonyms for the genes. Or if I select this column instead, I have a different set of properties that relate to the protein. For example, you know, a full list of, for example, what is its what is it? No, not much there. Delete this column. But I've essentially built up this smart table by adding comment, for example, like links, database links. I can say add all the links to any particular database that I might have, for example, the PFAM links, because I've already added the UniProt links. So I can build this up in various ways. So these are the properties. I can also do transforms.
Okay, yeah, why don't you do that, and then maybe show people how to, once you do that, maybe show people how to select the human database, because people are asking about the human database.
So I can, from the various transforms, for example, all the pathways of the gene, which I've already shown there, or all the regulators of the gene, for example. So I'm getting a set of regulators here. I can also see this, if I can also switch the parity. So if to create, for example, a smart table of pathways now, and these are the genes from my original smart table that match each of these pathways. So this essentially flips the parity. So I'm not quite sure what the question was about human. I can, if I want to create a new smart table, well, I can, if I select human. Well, actually, I'm not quite sure. What is the question, Peter?
Just go ahead and show them how to select humans. Because the idea is that when you select any other database, including humans, all of these same tools are available. So for example, you could show the human cellular overview. So all these same tools are available for humans as well as other organisms. The smart tables are available for humans.
Yeah, I wanted to actually show the special smart tables. So
right.
you have your own smart tables that you can create, and you can upload a file of things or create it from any query elsewhere on the site. But we also predefine all these special smart tables for every organism that we have, which contains a list of all objects of a particular type in this database. So if I want to look at, for example, all human pathways, I would go to this smart table. And now I can do all the same things. For example, I can say, what are the genes involved in this pathway or in each of these pathways? And it will show me. So I can do various things with this. Once I have a smart table, there are various things I can do with it. I can export this to a file on my machine as a spreadsheet, so I can look at it. If these are pathways, I can export them to a collage. If these are genes, I can export them to a FASTA file. I can also, if it's a data set that I can paint this data on the cellular overview. Now it's not going to be very useful if I'm painting all the pathways on the cellular overview, but if I have created a smart table that contains a subset of them, I can display them on the cellular overview. If my smart table contains data columns, which can happen if I can upload a smart table by uploading a file, and there I can have maybe genes in the first column and then a set of data columns elsewhere. So if I do that, I can use those as input to my various omics analysis tools.
I think at this point, we should probably hand things over to Jeremy.
Okay.
So let me introduce Jeremy Zucker, who is a staff scientist at Pacific Northwest National Lab. And we'll be talking about metabolic modeling using pathway tools for Neurospora crassa.
Thank you, Peter. I am going to share my screen right now. Cool. Can everyone see the screen?
Looks great. Okay,
good. So first of all, I just want to finish answering the question from Christiane Hassenrook, which is that there are 4 choices for multiple hypothesis correction in the enrichment analysis. And you have to specify whether it's spatial or temporal data with the columns that you include in the enrichment in the smart table that you analyze. I'll just go ahead and send that right now. Okay, so thank you, folks. I want to talk about really how pathway tools has helped me develop, help me analyze metabolic and regulatory networks of the circadian clock in Neurospora crassa. And just as a little background, Hang on for just a second. Just let me get my thing out of the way. Okay, so the filamentous fungus Neurospora crassa was first discovered as an orange mold infestation of Paris bakeries in 1843 and has since become a model organism for eukaryotic biology. Work on Neurospora has led to essential discoveries in circadian rhythms, epigenetics, genome defense, mitochondrial biology, post-transcriptional gene silencing, and DNA repair. Most famously, work in the 1940s by Beadle and Tatum led to the Nobel Prize-winning one gene, one enzyme hypothesis that established the fundamental link between genes and proteins in all organisms. Work on Neurospora thus paved the way for modern genetics and molecular biology. Of equal consequence, the work by Beadle and Tatum ushered in a new era in the study of cellular metabolism and phenotypes. The genetic facility of Neurospora, coupled with its ability to grow on minimal media, simplified the isolation of mutants with additional nutrient requirements. These first such auxotrophic mutants established the universal link amongst genes, enzymes, and the ordering of reactions in biosynthetic pathways. Work over subsequent decades has led to a compilation of hundreds of such mutants, shedding light on most major biosynthetic pathways. With the sequencing and annotation of the Neurospora genome, these genetic data could be organized on a physical scaffold. Genetic markers could be assigned to specific genes with predicted biochemical functions. Genes could be assigned to previously orphaned biochemical reactions, and a global map of Neurospora metabolism began to emerge. More than simply a catalog of reactions, a metabolic network can capture the genotype-phenotype relationship in an executable form. For example, if we provide the metabolic network with minimal media, and the network is capable of producing all of the essential compounds that are necessary for growth, then this is a prediction that the organism is capable of growth in that nutrient condition. If we then remove a reaction from the network, and one or more essential compounds can no longer be produced, then this is a prediction that the gene that codes for the enzyme that catalyzed that reaction is essential. If we supplement the minimal media with additional nutrients, and the mutant is now capable of producing all of the essential compounds necessary for growth, then this is a prediction of an auxotrophic mutant. The wealth of genetic and metabolic data available for Neurospora, along with efforts to knock out and phenotypically characterize all 10,000 genes in the genome, provides a strong foundation for the development of a genome scale metabolic model. A metabolic model would in turn complement experimental efforts by integrating data from experiments on single genes into a coherent genome-wide metabolic framework, providing potential mechanistic insight into experimental phenotypic observations and enabling the comprehensive modeling of perturbations that could not be feasibly performed in the lab. A genome scale model is also a requirement for the rational and efficient use of Neurospora as a potential biofuels organism. The process that we use to reconstruct the metabolic network of Neurospora can be broken into 4 stages. In the first stage, indicated in purple, the genome sequence annotation was used to automatically infer enzyme complexes, transporters, and reactions. Pathways were manually curated using experimental evidence from the Neurospora, reactions were used to predict metabolic pathways derived from BioCyc, and pathways were manually curated using experimental evidence from the Neurospora bibliome. Gene-protein reaction associations, pathways, and literature citations were used to construct the first draft of the Neurospora Psych pathway genome database. For the second stage, indicated in blue, we utilized a suite of algorithms we designed called FARM that suggests changes to the metabolic network based on a training set of experimentally confirmed growth phenotypes. These suggestions were reviewed manually and accepted into the final model if they were consistent with published experimental evidence. In the third stage, indicated in green, we independently validated the predictive accuracy of the final metabolic model on a test set of growth phenotype observations that were not utilized during model construction. In the fourth stage, we utilized the validated metabolic model to predict nutrient rescue for all oxytrophic mutants and to predict synthetic lethal interactions for all pairwise gene knockouts. And as I'll show later on, we also use this to analyze the circadian clock. So I now want to go into a bit more detail about how we use pathway-directed curation to construct the initial draft model. To predict enzyme function, we use the EFICAS2 enzyme prediction software, which takes PFAM domains, prosite patterns, and Swiss-Prot sequences, and generates an enzyme family-specific categorizer composed of hidden Markov models, functionally discriminating residues, and support vector machines, which were combined into a decision tree to arrive at an EC number prediction. Out of the 9,934 protein sequences, we assigned 808 genes to 1,023 unique enzyme activities. To curate protein complexes, we use the Pathway Tools enzyme complex editor, which identifies multiple proteins of a reaction and presents all possible combinations of complexes to the curator. Of the 291 biochemical reactions that were associated with multiple proteins, 47 were manually curated as being catalyzed by an enzyme complex, and we assume the rest were catalyzed by isozymes. We also use the transport inference parser, which filters protein annotations for transporters, infers multimeric complexes, the substrate, and the energy coupling mechanism. We took 9,934 free text protein annotations and identified 290 transport reactions that transport 137 metabolites between the cytosol and each organelle or extracellular space. To predict pathways, we use Pathologic, which computes the probability that the pathway exists in Neurospora by taking into account the number of reactions in a Metabolic pathway, the number of reactions in the pathway that are associated with an enzyme in Neurospora, and the number of reactions that are unique to that pathway. Of the 1,770 enzyme-catalyzed reactions, we assign 893 to 257 pathways. Biomass modules were separately defined for DNA, amino acids, cell wall, lipids, sterols, essential cofactors, and secondary metabolites. This modular decomposition allowed for different goals and different applications of the model. For example, we know that wild-type biomass contains substantial amounts of secondary metabolites, such as sphingolipids, ergosterol, and carotenoids, which gives Neurospora its characteristic orange color. So we included the secondary metabolites module in the biomass objective when predicting wild-type fluxes. On the other hand, secondary metabolites are not strictly required for viability, so we removed this module from the biomass objective when predicting gene essentiality. So what we ended up resulting is a metabolic map of Neurospora. And actually, to create this poster-sized figure, we generated organelle-specific pathway genome databases and then stitched them all together in Adobe Illustrator. But what we ended up getting was something that is really quite useful. So now what I want to talk about are some algorithms that we developed that use known phenotypes to help us curate our model. And I should mention that most of these algorithms are now part of BioCyc, so you can use them too as part of the Metaflux module. So to accelerate the prediction of phenotypes, we developed 3 optimization-based algorithms, which together comprise fast automated reconstruction of metabolism, FARM. These algorithms are LIMED-FBA, which predicts flux while linearly accounting for metabolite dilution, OnePrune, which removes blocked reactions with a single compact linear program, and CROP, which reconciles differences between in silico and experimental gene essentiality by suggesting reaction additions and deletions to the metabolic network. In the interest of time, I'm only going to describe the last algorithm, CROP. The evidence used to decide which reactions to add or remove is influenced by whether we had manually curated a reaction with experimental evidence from the literature, what pathways a reaction was part of, and whether these pathways were predicted to be in Neurospora. Also, the thermodynamic estimates of Gibbs free energy and probabilistic estimates of enzyme function. This evidence was mathematically integrated on a probabilistic scale to assign each reaction a weight. Our approach to derive weights in a disciplined manner was motivated by the statistical maximum a posteriori estimator. Thus, weights have a direct probabilistic interpretation. They depend on the product of the probabilities that the reaction is biochemically and thermodynamically plausible. These weights guided crops' growth reconciliation towards metabolic network changes that were most consistent with available evidence. To achieve consistency when the model incorrectly predicts growth, crop suggests reactions from the model to remove. To do this, crop applies a mixed integer linear program, or MILP. To achieve consistency when the model incorrectly predicts no growth, crop suggests reactions to add from Metacyc. To do this, crop applies a linear relaxation of MILP. These suggestions were reviewed manually and accepted into the final model if they were consistent with published experimental data. Using this approach, we were able to recover 47% of all enzyme-catalyzed reactions with 491 organism-specific citations, which is comparable to other highly curated models. So now that we have a model, how good is it? To answer this question, we first curated a collection of 450 mutant viability phenotype observations on minimal media. We then separated the collection into a training set where knowledge of the viability phenotype was used to improve the model, and a test set where the viability phenotype was hidden from the model. Accuracy was measured as two quantities, sensitivity and specificity. Sensitivity was defined as the proportion of experimentally viable mutant observations that were predicted to be viable in silico. And specificity was defined as the proportion of experimentally inviable mutant observations that were also predicted to be inviable in silico. On the training set, we correctly predicted growth in 107 out of 108 gene knockouts that were experimentally observed to be viable. And we correctly predicted no growth in 44 out of 47 mutants that were experimentally observed to be inviable under minimal nutrient conditions. On the test set, we correctly predicted 270 out of 289 experimentally viable gene knockouts, and we correctly predicted 13 out of 14 experimentally lethal mutants. To predict rescue phenotypes, we first curated a collection of 96 nutrient conditions in which an auxotroph was rescued when minimal media was supplemented with a nutrient. We then separated the collection into a training set where knowledge of the rescue phenotype was used to improve the model, and a test set where the rescue phenotype was hidden from the model. But because we only collected data on which nutrients rescued the auxotrophs, we could only measure sensitivity, but not specificity. On the training set, we correctly predicted 74 out of 77 rescue phenotypes, and on the test set, we correctly predicted 17 out of 19 rescue phenotypes. So what was the underlying cause of these errors? In the training set, the only auxotrophs we were unable to correctly rescue were due to condition-specific regulation that our model does not capture. So according to experimental observation, mutants in ACE2, ACE3, and ACE4 can grow in acetate minimal media only because the enzymes in the glyoxysome are induced when extracellular acetate is present in the medium. Conversely, ACE2, ACE3, and ACE4 mutants cannot grow in sucrose minimal media, even though sucrose can be converted to acetate via pyruvate fermentation, because the glyoxysomal enzymes are not expressed in this condition. Because the acetate-dependent induction of the glyoxysome is not included in our model, we could not successfully predict both inviability under sucrose minimal media conditions and viability under acetate minimal media conditions. Our model also does not account for regulation of enzyme activity. An illustrative example is GLIN1 and GLIN2, which code for the alpha and beta subunit, respectively, of glutamine synthetase. Our model requires both subunits for enzyme catalysis. However, it was experimentally shown that concentration of extracellular ammonium regulates this enzyme subunit composition, which can include both subunits, only alpha subunits, or only beta subunits. So this metabolic complexity highlights the need for future incorporation of kinetics and regulation. In one instance, however, a prediction initially thought to be an error provided the means to identify an issue with an experimentally observed knockout. The viability phenotype for ERG14 was observed on a knockout strain originally designated as a homokaryon, and experimental observations of the strain revealed a normal growth phenotype. In contrast, our model predicted that the ERG14 mutant was blocked in the production of mevalonate, which is a necessary precursor for the sterol component of biomass. Moreover, previous efforts to phenotype temperature-sensitive mutants of ERG14 revealed severe morphological defects, as shown in the image. These defects were expected to be lethal in the full knockout. So driven by these inconsistencies, a re-examination of the ERG14 knockout revealed that the mutant used was in fact a heterokaryon and not a homokaryon as it was initially designated. This prediction, in effect, served as a blind control that highlighted the predictive power of the model. So now I just want to shift gears a little bit so that we can talk about some of the metabolic and regulatory investigations into the circadian clock, now that we have a model that we actually believe in. So transcriptional and translational feedback loops in fungi and animals drive circadian rhythms and protein levels, but post-transcriptional mechanisms also contribute. So to determine the extent and underlying source of this regulation, we applied newly developed analytical tools to a long duration, deeply sampled circadian proteomics time course comprising half of the proteome. We found a quarter of expressed proteins are clock regulated, but more than 40% of these do not arise from clock regulated transcripts. And our analysis predicts that these protein rhythms arise from oscillations in translational rates. Our data highlighted the impact of the clock on metabolic regulation, with central carbon metabolism reflecting both transcriptional and post-transcriptional control, and opposing metabolic pathways showing peak activities at different times of day. The transcription factor CSP1 also plays a role in this metabolic regulation, contributing to the rhythmicity and phase of clock-regulated proteins. So just to show you how much of the metabolic network is circadian, down here on the bottom, we see that these colored arrows here correspond to the color key down here, denoting peak phases denoting peak phase timing of rhythmic enzymes. So the idea is that we have, and this is circadian time, so which is actually 22.5 hours, which makes a full circadian clock. And what we see here is that different enzymes are circadian, but they peak at different times during the day. And so we're just looking at the full distribution of which proteins are circadian and when they peak. Perhaps a more informative analysis is what happens when we take a look at the proteins that are rhythmic in the pentose phosphate pathway that peak in antiphase to those in glycolysis. So enzymes within the pentose phosphate pathway are shown here on the left, and enzymes within glycolysis are shown here on the right. And bolded and colored arrows denote enzymes that are considered rhythmic in our analysis. And the colored denotes how many hours away it is from peak abundance. So at CT12, which is circadian dusk, then it appears that glycolysis is at a peak, but pentose phosphate is at a trough. And so we could actually take a look at a movie that shows how the metabolic enzymes shift shift in antiphase to each other. So you can see at circadian time 12 at dusk, we have the strong glycolysis pathway, but as the time moves on, then now at circadian time 24, we have pentose phosphate taking over and glycolysis disappearing, and then returning back again to glycolysis at 24 hours or at dusk again. So let's see, next slide. So another way of looking at this is was inspired by the omics dashboard. So this is the average modeled rhythmic mRNA and protein levels within BioCyc categories that suggest post-transcriptional regulation of metabolic pathways. And so what you're looking at here is mRNA is represented always in orange, and protein expression is measured in blue. And so we can see that not only is glycolysis out of phase with pentose phosphate, but also the mRNA expression is out of phase with the protein expression, but not in every case. In some cases, they're out of phase, and in some cases, they're in phase. And so we can see, we can look at, and this is also, because this is the omics dashboard, this is the this is the average number of, this is the average expression of each gene in each pathway. And so, and then the shaded regions show the distribution. Now we can actually look at a more dashboard centric view of by comparing rhythmic mRNA and proteins within many different kinds of BioCyc pathways. And in this case, the little small dots represent the individual gene expressions or protein expressions, and the central dots indicate their average. And so again, we can see that, so for example, with amine synthesis, they are in phase with each other, but with carbohydrate synthesis, they're 180 degrees out of phase with each other. And in other cases, they are they're slightly out of phase with each other. And so it was interesting to analyze these their circadian rhythms from this perspective. And then the last figure I want to show you is differences in rhythmicity of proteins between wild type and CSP1. So CSP1 is known to be important for metabolic regulation, and what it appears to make a difference is that it changes the phase at which the circadian clock occurs. And so it's still circadian after you knock out CSP1, but sorry, I don't know why that flipped forward. But it still continues. But so even though it's still circadian, the phase is shifted as a result across many, but not all of the different metabolic pathways. So with that, I just want to acknowledge my colleague, Bill Cannon and Natalie Munoz at PNNL, James Gallagher and Jonathan Dreyfus at BU, my co-author and now wife, Linda Ocasio-Zucker at Tardigrade Biotechnologies, the folks at Texas A&M who performed the biolog analysis, which I didn't have really time to talk about today, the folks at Dartmouth and RPI who performed the proteomics analysis, the pathway curator, Heather Hood at OHSU, the folks at UC Riverside who performed almost all the gene knockouts, Alan Radford at the University of Leeds, who maintains the Neurospora e-compendium from which we obtain the mutant information, Peter Karp, of course, and the rest of the folks at SRI who developed MetaPsych and the Pathway Tools software that lies behind Neurospora Psych, and the genome annotation informatics folks at the Broad Institute who worked diligently to make sure the genome annotation was far better than it was. So thank you for your attention, and now happy to take any questions. So it looks like.
Thank you for a great talk, Jeremy. Did you put up the URL for your Neurospora website?
I didn't. It's currently not available, but I will. But I will go ahead and put a link in the chat window right now, just so that you have it. It's psych.pnnl.gov. Okay,
so sorry about that. I didn't realize that. Great, and when we do have a few questions, but we've got some time for questions here.
Good.
How do you maintain constant nutrient influx during circadian cultivation to avoid starvation?
That's a great question. So I believe what we did is that we started them. Well, when we're doing, this actually becomes really important when we're doing metabolic flux analysis, right? Because we want to make sure that there is enough glucose for us to do 13C tracer analysis. And if for that, what we did was we put them in a conidial state. So they're basically just like, they're just, it's mostly just, it's just tiny little conidia. And then by the time they grow up, before they get filamentous, is when we're actually performing all the measurements. So in that case, there's plenty of glucose and it doesn't run out before we finish the glucose 13C analysis. I'm actually not sure how they do it, how they actually perform the analysis. But I know that you can actually run these race tubes and you can see how the circadian clock will along these race tubes because there's a specific conidiation stage. And so you get these bands of orange as the circadian clock runs along. And you can see this in these race tubes where time is basically the vertical axis.
Great. Another question. It looks like you observe circadian cycle, but cannot explain it mechanistically, correct?
Oh, so we have a mechanistic explanation of it. We were more now teasing apart exactly what so you're right. So this multi-omics analysis was just observing that that there were these circadian clocks. There are other parts of the paper that I didn't describe, which basically teased apart the hypotheses. And so the question is, could it be protein degradation that's causing protein circadian? So one thing that we observed is that there are many cases where mRNA was not cyclic, but the protein was cyclic. And so the two hypotheses is that it could either be post-transcriptional regulation or it could be protein degradation. And at least in Neurospora, it appears to be the case that it's post-transcriptional regulation that drives the protein circadian clock and not cyclic degradation. And we provided a number of different kinds of evidence to support that hypothesis.
Okay. In your circadian study, do you expose microorganisms to the light dark cycle?
So the way it works, at least in terms of this analysis, and there's different ways of doing it. So one way of running this experiment is you keep it in the dark and then you shine a light, and it has a blue light sensor that basically resets the clock. And so then the phase begins at CT0. And then by the time dusk reaches, that's when we're interested in that seems to be where all the protein expression is occurring. The other way of doing it is to so that's the main way that it works is that we shine a light on it, and that's CT0, and then it continues from there, and that's how we analyze these networks. Did that answer your question?
Not sure. Another question is, fungi form spores. Can you model this stationary phase of their development?
Oh, that's a great question. So what we would need in order to do that is expression data from different stages of the fungi, and then we could see which enzymes are expressed or not expressed at different times of the day, at different stages of growth. I don't have that data, or I haven't looked at that data. I've mainly been focused on circadian and also just that initial stage where the light gets turned on. But there are folks who are working on kind of other questions about Neurospora that would be able to answer those kinds of questions with that kind of data.
Okay, I think that's it for questions on your talk, Jeremy. There's still, so thank you very much. Great presentation.
Sure. It looks like we got 138 questions answered, which is pretty impressive, and 14 remaining. So maybe we'll, but I think we're probably out of time at this point. Is that where we're at?
Well, we still have 9 minutes actually. So I think we can go through some of these other questions. Like one is, can metatranscriptomic data be integrated? You can display metatranscriptomic data for a microbial community or other community using multiple metabolic network diagrams, as I showed in a slide in my talk. where essentially you can build a PGDB for each organism in the community. Okay, so here are the overall steps is you take a metagenomic data set, you bin it into separate organisms using various software tools that exist. You then get a separate MAG or metagenome assembled genome for many of the organisms in the community. Then you feed each MAG into pathway tools and create a separate PGDB for that organism. You now have this family of PGDBs describing all the organisms in the community. We have a number of tools that can operate on a set of PGDBs. For example, you can issue searches across all the MAGs in a set to find what genes do they contain, which organism has a given gene or a given pathway or a given metabolite. And you can create cellular overview diagrams, metabolic network diagrams for each MAG, each organism in the community. You can then overlay metatranscriptomics data onto that family of metabolic network diagrams to look at the activation levels of pathways across all the organisms in the community. Okay, actually another question for Jeremy. I was curious about the out of sync mRNA. There were a few proteins that peaked about when their mRNA troughed. What do you make of these?
Yeah, that's a really good question, right? So it's one could understand, let's suppose there was no protein degradation, right? Then in that case, essentially proteins are the integral of the mRNA signal. So you would expect that if you have a cosine wave, that it would be what, negative sine, right? And so that can explain the shift. It just takes a while for proteins to protein accumulation to occur after due to translation, but this is like several hours away, right? So especially in cases where they're completely out of phase, like anti-phase to each other, then the integral is not sufficient to explain that difference. And so there's something else going on there, and I'm not really sure what it is, but it's very curious for sure. The other thing is that these are average phases, not individual phases. So it may still be the case that a given, now it's hard to say why. So that I think that's an area for potential future investigation.
Then there was a question about what I just said about metagenome PGDBs. They're asking, how do we arrange the bins of different organisms in a sample? You basically just create a separate directory, a separate folder where each folder contains data from one organism. And you can manually run those through Pathologic, or you can use our batch command line version of Pathologic to process each organism in turn. And each time you invoke it from the command line, you give it a different directory as the source of the data. What if the sample has been collected from an unexplored site? Not sure I understand that part, but the sites doesn't really matter to the software. It will just process the genomes that you provide.
Oh, there's one from Kemalal. Oh, sorry. Go ahead, Peter. I apologize.
I didn't actually have one to go next, but if you want to answer, go ahead.
Yeah, there's one from Kemalal near Malakar who says, does Proca GenBank file have abundance values of pathways and genes? I want to use that as an input file. So that's an interesting question, because certainly with metagenomes, you can do that, right? You can, there's a feature in Pathway Tools that takes not only the gene, but also the number of times that a particular function appears. And so you can use that information to build these metagenomes. Is that right, Peter?
I'm sorry, I was reading some of the other questions, so I didn't quite follow that.
So the question is, when you have a metagenome, right, where you can have like thousands of genes with a specific function, isn't there a function in or isn't there a file slot within the file format that allows you to specify how many times a particular function exists in your metagenome?
Yeah, there is. There is. Now that is in the pathologic format, which is yet another input format. And I was trying to remember if we support abundance values in GenBank format. I think we do, but I'm not positive. You'd have to check the software user guide or send us an email. I did give our support email address, so you all are welcome to email us questions at the support address that I gave. And there were a couple questions about combining metabolomics and transcriptomics data together.
Mhm.
In general, that's one of our future directions to improve the tools in that area, but you can combine them in one input data file now. You would simply have transcriptomics data has a gene in the first column, a gene name or identifier, and metabolomics data would have a metabolite name or identifier in the first column. And the tricky issue is that because the color, there's only one color mapping. In other words, the mapping of data value ranges to colors, there's only one such mapping. So the values for the metabolomics and the transcriptomics data have to be either in the same space, or I suppose you could actually partition them so that half the color mapping was used for metabolomics and half was used for transcriptomics. You could set it up that way as well. We haven't really explored that, but that's the basic approach. Okay, can we use the string database to create a pathway or network in BioCyc? Not that I know of. We haven't really set up any explicit connection there.
I want to insert, Javiera Menezes says, I want to insert a pathway in E. coli that it doesn't have. So I understand I have to identify the genes for the enzymes in the pathway that produce the compound I want, but I don't understand how the process is exactly. What tools would help to do this, create a pathway network, also model how would the production of that pathway behave? Can you give me some orientation of the process, please? Sure. So for any other pathway genome database except for E. coli, they're editable. And so if you have so I work in Pseudomonas petita, and so it's very so the desktop version of Pathway Tools makes the PGDBs editable, and so you can very easily import new pathways into your organism of interest. With E. coli, it's a little different because it's a read-only database, mainly for efficiency purposes. But I think it's actually possible to save as a PJC E. coli. You can take E. coli and save as, and then you create an editable version of that E. coli pathway genome database that then you could insert new pathways into. Now, in terms of how you can use that to model, so as far as the pathway model is concerned, you just have a bag of enzymes, and so if you've added new enzymes and you also have to have new reactions associated with those enzymes. So as long as you have that new pathway in the model, then when you run flux balance analysis, then it will just utilize that additional pathway. Now, typically, the objective for an organism is to grow as quickly as possible and not to produce your compound of interest. And so you could change the objective function to try to maximize the growth rate, or to not maximize growth rate, to maximize the flux through that pathway. And that would answer your question as to an upper bound for the titers, rates, and yield for that specific bioproduct. But then the real challenge comes into how do I sculpt the metabolic network so that in order to grow, I must produce some of this compound. And that's a separate tutorial on cell factory design that we can also give.